Exploring STR in human. Variable number tandem repeats, VNTRs, show high variation in the number of repeats in the population and are commonly used in forensics, for example, DNA fingerprinting, and to study genetic diversity. Short tandem repeats, STRs, also known as microsatellites, are an example of VNTRs. Part A. Go to the region from 307.4.666 to 307.5.1.0.0 base pairs on human chromosome 4. Which gene does it overlap? Which exon of this gene falls in this region? We're first going to start from Ensemble homepage, and we're going to search for our location on the search bar in the center of the page. We can either type in human space 4 colon our start coordinate, hyphen, or end coordinate. Alternatively, we can also select our species of interest by this drop-down menu here. Now that I've specified my species and my genomic location, I'm now going to click Go. We're now on the location page. This page shows us our region in detail. We can find information about the gene falling in our region of interest by focusing on Region in Detail view. The red box indicates our genomic location. And from this diagram, we can see that our region of interest overlaps with the HTT gene. We can also find more information about the overlapping transcript by scrolling down to the zoomed-in version of our region of interest. We can find out information about the exon of this gene that falls within this region by clicking on this golden transcript. The pop-up box gives us information about the exon. We can see it's exon 1 of 67 of the Huntington gene. Part B. Configure this page to turn on repeats, low, simple repeats, and tandem repeats tracks in this view. Can you see any repeats in this exon? What tools were used to annotate the repeats according to the track information? To answer this part of the exercise, we're going to click on this blue button which says Configure this page. We now have a list of all the tracks available, and we're now going to search for our tracks by searching for them in the search box. We're now going to turn on the tracks that the exercise mentions, which are repeats, low, tandem repeats, and simple repeats. Once we're happy with our track selection, we're going to save and close our configurations. Once our tracks have loaded, we will find them at the bottom of our region in detail view. We have our repeats, simple repeats, and tandem repeats. There are two types of repeats in this exon. We can find more information about these repeats by clicking on these bars. We have polyglutamine polyQ tract, or CAG, and we also have polyproline polyP tract, or CCG. To find out more information about our tracks, we can simply click on their name and click on the eye icon. The Tandem Repeats Finder was used for this annotation. And if we click on Simple Repeats, we can see that Repeat Masker was used to find repeats and low complexity sequences. Part C. Zoom in on polyglutamine, polyQ tract, or CAG to see its sequence. How many CAG repeats can you see in the human reference assembly? Does this track overlap any phenotype-associated variants? What is the identifier of this variant? To zoom in on the polyglutamine polyQ tract, or CAG, I'm going to draw along the tract, and I'm going to jump to the region. We've now zoomed in on our polyglutamine polyQ tract or CAG repeat. We can find the number of CAG repeats in the human reference assembly in the sequence track above the contigs. There are 19 CAG repeats in the human reference sequence. These CAG repeats also overlap a phenotype-associated short variant, which is indicated by the pink bar. We can find the RS ID of this variant by clicking on this pink bar. The 19 CAG repeats in the human reference sequence overlap RS 711-80116 variant. Part D. Go to the Variant tab of the phenotype-associated variant. What is the consequence ontology of this variant? Does the reference allele match the number of repeats you have just counted? What is the shortest and longest allele? To find more information about this variant, we're going to click on the pink bar and we're going to navigate to the Variant tab by clicking on this RS ID. We're now on the Variant tab. The Variant tab has a summary of this variant. We have information about the most severe consequence, which is in-frame insertion. We also have information about the alleles associated with this variant. To show all the alleles associated with our variant, we're going to click on Genes and Regulation on the left-hand side. This variant has many alternative alleles which differ in the number of repeats. If we scroll across the Genes and Regulation table to find the codons column, we can find that the first allele in the codons column in the Genes and Regulation table is a reference allele. It is also composed of 19 CAG repeats, just as we saw in the Region in Detail view. In Ensemble, alleles are given as reference/alternative allele. In our codons column, we can also find the reference allele / the alternative alleles. Alternatively, we can also find information about the alleles in the variant summary in the alleles line. We can show all the alleles in the summary page by clicking on this plus icon here. From this allele information, we can find our shortest allele and our longest allele and count the CAG repeats within these. Part A. Are there any phenotypes associated with this variant? To find information about the phenotypes, we're going to click on Phenotype Data on the left-hand side. This variant is associated with Huntington disease. This association was made by ClinVar, and we can find the supporting evidence by clicking on these PubMed ID links.